It's time for the 3-question warm-up for Immunology 3. Let's get going. What muscles and nerves are derived from the third branchial arch? So this is the stylopharyngeus muscle, which is innervated by the glossopharyngeal nerve, cranial nerve 9. Next question. What drug category is often used in the treatment of myasthenia gravis? So we use acetylcholinesterase inhibitors like pyridostigmine and physostigmine to treat myasthenia. Next, to which class of antiarrhythmics do each of the following agents belong? So first we have sotalol. Technically it's a beta blocker, but its strongest action is as a potassium channel blocker, so it's classified as a class 3 antiarrhythmic, so don't get fooled by sotalol. Propranolol is a typical beta blocker, and it's class 2. Bertilium is a class 3, and quinidine is a class 1. That's it for the warm-up. Let's get to that lecture.
We've talked about how the antigen-presenting cells like dendritic cells capture antigen and then present it to helper T cells in order to activate those T cells to fight pathogens. So in this video we're going to look at some deeper details about T cells. So where did T cells come from? Well, the T cell precursors originate in the bone marrow, but they mature in the thymus. Remember, the T in T cell is for thymus. And make sure you remember that the thymus is derived from the epithelium of the third branchial pouch. The thymus is sort of like finishing school for T cells. It's where T cells go to develop and differentiate and mature. I want you to think about the thymus as a circle, and then within that circle is another circle. So the outer circle is the cortex of the thymus, and the inner circle is the medulla of the thymus. Now, the cortex is where immature T cell precursors start out, and as they mature, they gradually move toward the medulla. So mature T cells are found in the medulla of the thymus. So remember, M for medulla and M for mature T cells. And then as they move from the cortex toward the medulla, a very important process called selection is taking place, where you're selecting for T cells that you want to keep and removing T cells that you don't want to keep. So how exactly does this differentiation and selection work? Well, the T cells in the cortex of the thymus are immature and unspecialized, and they have both CD4 and CD8 surface markers. They also have T cell receptors, which can bind to antigens and MHC molecules. And the cortex of the thymus displays a bunch of different antigens on MHC1 and MHC2, and it's basically looking for which immature T cells have a T cell receptor that's capable of binding to MHC. Not too strong, not too weak, just right. So if they bind too weakly or too strongly, the thymus is going to let them die. But if the T cell can bind just right, the thymus is going to feed it a survival signal, which is going to keep it alive. And then, still in the cortex, the T cell precursors that bind really well to MHC1 are going to lose their CD4 marker, and they're designated to eventually become a CD8 positive cytotoxic T cell. And the T cell precursors that bind really well to MHC2 are going to lose their CD8 marker, and they eventually become a CD4 positive helper T cell. And that entire process of selecting the T cells that are capable of binding to MHC is called positive selection, and that takes place in the cortex. Then negative selection takes place at the corticomedullary junction, which is the border or junction between the cortex and the medulla. So negative selection is where you're selecting out T cells with receptors that bind to self-antigens. So when your thymus finds a T cell that wants to bind to self-antigens and attack your own body, the T cell is selected out and destroyed by apoptosis so that you don't have an autoimmune phenomenon. Only about 2% of the T cell precursors in the thymus actually survive this positive and negative selection process. So most of them get killed off, but the ones that do survive eventually leave the thymus as naive T cells. But that's not the whole story, because once those CD4 positive helper T cells are activated, they can become either a T helper 1 cell or a T helper 2 cell, which have completely different functions. T helper 1 cells stimulate macrophages and they stimulate cytotoxic T cells. T helper 2 cells stimulate plasma cells to make antibodies. So let's look at how this differentiation takes place. When a naive helper T cell leaves the thymus, it's a T helper 0 cell. Now if IL-12 is around, say during a viral infection, IL-12 will induce those naive T cells to become T helper 1 cells. Now, why do you want more T helper 1 cells around during viral infections? Well, let's talk about which cytokines T helper 1 cells produce. T helper 1 cells secrete IL-2. What does IL-2 do? Well, IL-2 stimulates proliferation of T cells, including the cytotoxic T cells that are going to destroy virus-infected cells, as well as stimulating proliferation of even more of these T helper 1 cells. So T helper 1 cells also secrete interferon gamma, which activates macrophages to go eat up the invading bacteria or whatever it is. And interferon gamma also suppresses proliferation of T helper 2 cells. So You need to remember that T helper 1 cells secrete IL-2 and interferon gamma, which is going to stimulate cytotoxic T cells and stimulate macrophages. Then there's a third substance to know in association with T helper 1, which is IL-10. IL-10 is an inhibitory interleukin. It inhibits proliferation of T helper 1 cells. But if you go back to that initial naive T helper 0 cell, what if instead of IL-12 there was IL-4 around? Well, IL-4 induces T helper 0 cells to differentiate into T helper 2 cells. So what cytokines do T helper 2 cells make? They make IL-4 and IL-5, which is going to stimulate proliferation of B cells. And they also make IL-10. What did we just say IL-10 does? It inhibits T helper 1 cells. So if you're making T helper 2 cells, they're going to pump out IL-4, which stimulates the production of more T helper 2 cells, and they're going to pump out IL-10, which inhibits T helper 1 cells. So you're either going to get more T helper 1 cells or more T helper 2 cells. It doesn't make sense to produce both at the same time. And then IL-10 also inhibits macrophages. So T helper 2 cells are pro-B cell and anti-T helper 1 cell. So make sure you know those different cytokines and these T cell differentiation pathways. Now, before a helper T cell can differentiate into a T helper 1 or T helper 2 cell, it has to be activated. And what does it take to activate one of these naive T helper 0 cells? Well, it takes an antigen-presenting cell to present it some antigen. You don't want T cells to be activated until there's something to attack. So a T cell precursor in the thymus goes through the selection process and then ends up as a CD4 positive T helper 0, a naive T helper cell. And then it migrates out to the lymph nodes and just hangs out until a dendritic cell comes galloping in from the front lines with some antigen to present to the lymphocytes that are in that lymph node. And regardless of whether you're a T cell or a B cell, lymphocytes need not one but 2 signals to be activated. It's kind of a system of checks and balances to make sure that you don't accidentally activate them. Now the first signal is that antigen that the dendritic cell is presenting. Then the second signal is a co-stimulatory signal. On helper T cells, you have a specific T cell receptor plus a CD4 surface marker. And that T cell receptor recognizes an antigen that's presented by the MHC2 molecule on the antigen-presenting cell. And remember we said that antigen-presenting cells like dendritic cells also have a B7 protein on their surfaces. That's involved with this second signal, which is called a co-stimulatory signal. And remember that B7 is kind of a family name. The specific B7 protein can either be a CD80 or CD86. But either way, that B7 protein on the antigen-presenting cell is going to bind to a CD28 surface marker on the T cell, and that's going to activate that helper T cell to start producing cytokines. If it's a T helper 2 cell, it's going to start producing IL-4, IL-5, and IL-10 in order to stimulate B cells and to suppress T helper 1 cells. Or if this is a T helper 1 cell, then when it's activated, it's going to start producing interferon gamma and IL-2. Now, what about cytotoxic T cells? How do they get activated? Well, cytotoxic T cells are looking for virus-infected cells to kill, right? Or cancer cells to kill. So these cytotoxic T cells have a T cell receptor, but again, instead of that CD4, they have CD8. And that TCR CD8 complex is going to recognize an antigen that's presented on an MHC1 molecule. So CD8 recognizes MHC1, and that's your first signal. And then the second signal is that, again, the T cell has a CD28 surface marker that recognizes a B7 protein on the antigen-presenting cell. And then once that cytotoxic T cell has been activated, it can go kill infected cells. And also, the IL-2 that the T cells are making is going to stimulate clonal expansion of more of these cells with this specific T cell receptor, which allows the body to kill even more infected cells. Now, while we're talking about lymphocyte activation, let's go ahead and talk about how to activate B lymphocytes in order to make antibodies. Now here there are 4 main steps. The first step is that a helper T cell gets activated, and we just covered that. The next step is that the B cell endocytoses an antigen and presents it to the helper T cell. So remember, B cells can behave like antigen-presenting cells, so they're capable of bringing in antigen. The antigen binds to a surface immunoglobulin, and then the B cell swallows it up and digests it, put a little piece of that antigen on an MHC2 molecule on the B cell surface so that it can present the antigen to the T helper cell. So the T cell is going to recognize the antigen with its T cell receptor. The third step is another co-stimulatory signal. The B cell has a CD40 marker on its surface, which binds to a protein on the T cell called a CD40 ligand. And then finally, the first three steps have now induced this T helper 2 cell to produce IL-4 and IL-5, which stimulates proliferation of B cells and immunoglobulin class switching of the B cells. We're going to talk about the functions of B cells in another video. But in a sense, the B cell engulfs the antigen, then asks the T helper 2 cell for permission to fight the antigen. So that's pretty much it for the helper T cells. They help by secreting lots of cytokines to recruit other cells like macrophages, or they help by promoting B cells to proliferate and make more antibodies. Helper T cells don't do any of the actual killing, but they call in reinforcements and direct the battle. Now, before we talk about the other kinds of T cells, let's do a quick whiteboard review to go over the actions of these cytokines that are produced by and that regulate helper T cells. One more time. IL-2 stimulates proliferation of T cells. IL-2 also helps T cells differentiate into CD4 positive helper T cells or CD8 positive cytotoxic T cells or regulatory T cells, which we'll go over shortly. IL-4 stimulates proliferation of B cells. It also stimulates T helper 0 cells to become T helper 2 cells. IL-5 stimulates proliferation of B cells. IL-10 is an inhibitory cytokine, so it inhibits T helper 1 cells and it inhibits macrophages. IL-12 is what stimulates differentiation of T helper 0 cells to become T helper 1 cells. And interferon gamma activates macrophages and it also inhibits the proliferation of T helper 2 cells. And then what produces what? T helper 1 cells produce IL-2 and interferon gamma mainly. They produce other cytokines like IL-3, but the two main ones I want you to know are IL-2 and interferon gamma. For T helper 2 cells, I want you to know that T helper 2 cells produce IL-4, IL-5, and IL-10. And then just for completeness, IL-12 is produced by the macrophages and also dendritic cells. Good job on that. And for your prize, I'm going to give you a smiley face made out of stars. So in the thymus, T cell precursors are selected based on their T cell receptors to either become CD4 positive helper T cells or CD8 positive cytotoxic T cells. And if the helper T cells are in charge of directing the battle and recruiting cells to come and fight, cytotoxic T cells are more like the frontline foot soldiers, the ones that are actually fighting the battle, killing the enemy. Sometimes these are called killer T cells. Again, these are CD8 positive T cells, so they can bind to MHC1 on self cells, they can kill virus-infected cells, they can kill cancer cells, and they can potentially kill non-self human cells from a transplant. So, how do these killer T cells actually kill these enemy cells? Well, they do it by inducing apoptosis through a couple of different mechanisms. One mechanism is that cytotoxic T cells release granules of preformed proteins into the cells, including things like perforin and granzyme. So you can think of these like little grenades that damage the cells. Perforin perforates the plasma membrane, and then granzyme is a protease that activates apoptosis within the target cell. A second mechanism for cytotoxic T cells to induce apoptosis is that the T cell expresses a molecule called Fas ligand on its surface, and this Fas ligand interacts with a Fas receptor on the target cell, which triggers a series of downstream events which ultimately leads to apoptosis of that target cell. Another type of cell I want to mention are the natural killer cells, or NK cells. Now, NK cells are also lymphocytes, and they're kind of like cytotoxic T cells in that they also kill other cells, and they use perforin and granzymes to induce apoptosis. But whereas those true T cells are part of the adaptive immune system, with very specific receptors against very specific antibodies, the NK cells are part of the non-specific innate immune system, that more primitive immune system. Unlike the cytotoxic T cells, NK cells aren't looking for antigens that are presented on an MHC1 molecule. Instead, NK cells induce apoptosis in cells that do not express MHC class I on their surface at all. Remember, all nucleated cells are supposed to be displaying self-antigens on MHC1. And if they don't display those self-antigens, the NK cell can come up and kill those cells. So that's useful because sometimes virus-infected cells and tumor cells will actually down-regulate their expression of MHC1, almost like they're hiding from the cytotoxic T cells. But these NK cells are going to hunt them down anyway. Now, NK cell activity is enhanced by IL-12, by IL-2, and also by interferon alpha and interferon beta. Interferons are very important for fighting off viral infections. Not only do they induce natural killer cells, but they also induce nearby cells to inhibit viral protein synthesis. So if a cell is infected with a virus, it's going to start releasing interferon beta and interferon alpha, and those interferons are going to tell surrounding nearby cells, look, there's virus around. So you need to start generating these substances that are going to inhibit viral protein synthesis so that you can be protected from the virus. NK cells also secrete interferon gamma, which activates macrophages, like we saw with the T helper 1 cells. And NK cells can be identified by the presence of surface markers, CD16 and CD56. Now, I want you to know that CD16 can bind to antibodies down in the constant region of the antibody. And CD16 is found on the surfaces of NK cells, on macrophages, monocytes, and also neutrophils. So if an enemy cell gets coated with IgG antibodies, NK cells and these other cells with the CD16 surface marker are able to recognize antibody-coated cells and then kill them. And we call this process antibody-dependent cell-mediated cytotoxicity, or ADCC. There are two more types of T cells that I want to mention just in passing because they're not very high yield for step 1. Memory T cells and regulatory T cells. Memory T cells are T cells that don't go off to battle. They sort of hang back in the spleen and in the lymph nodes, and they kind of go into a dormant, inactive state so that if the body encounters that same pathogen a second time down the road, you have this subset of T cells that have already been activated against that pathogen, and they can be recruited to fight off the pathogen much more quickly the second or third time around. Now, memory T cells can survive a long time, and in the case of memory cytotoxic T cells, this is what allows some vaccines, especially live virus vaccines, to induce lifelong immunity, because that population of memory T cells can stick around for so long. Other types of vaccines may require boosters because they only induce memory helper T cells and sometimes memory B cells. We're going to go over all that again when we talk about vaccines in Immunology 5.
And then regulatory T cells are really poorly understood. Regulatory T cells are basically responsible for regulating the immune system by suppressing it. They inhibit B cells from producing antibodies. They inhibit helper T cells and cytotoxic T cells from doing their jobs. They produce IL-10, which is that inhibitory cytokine, and they release anti-inflammatory cytokines. If your regulatory T cells don't work properly and your immune system isn't suppressed when it ought to be, you're going to develop autoimmunity and atopic diseases where the immune system overreacts to benign, harmless antigens. So one of the picky details that's been known to turn up on step 1 has to do with a regulatory protein called forkhead box protein P3, or FOXP3. FOXP3 is a protein encoded by a gene found on the X chromosome, and this FOXP3 protein is a transcription factor that helps to control the development and function of regulatory T cells. Patients with a mutation in this FOXP3 gene can't suppress their immune system, and they develop a rare but lethal condition called IPEX syndrome. IPEX syndrome stands for immune dysregulation, polyendocrinopathy, like type 1 diabetes or thyroiditis, enteropathy, which leads to severe diarrhea and failure to thrive, and then the X is for X-linked. So the classic presentation is a male infant, because it's X-linked, who has severe chronic diarrhea, neonatal diabetes or thyroiditis, and eczema, atopic dermatitis. That's the immune dysregulation. So that's it for the T cells. Now I want you to pause the video and complete the end of session quiz, and then we'll go through the answers together.
All right, first question. Which cytokines are produced by T helper 1 cells and which cytokines are produced by T helper 2 cells? So T helper 1 cells produce IL-2 and interferon gamma, which leads to stimulation of T cells and macrophages. And then T helper 2 cells produce IL-4 and IL-5, which leads to activation of B cells and antibody production. And it also produces IL-10, which inhibits T helper 1 cells. Next, which cytokines inhibit T helper 1 cells and which ones inhibit T helper 2 cells? So we said IL-10 inhibits T helper 1 cells and interferon gamma inhibits T helper 2 cells. Next, how do cytotoxic T cells kill virus-infected cells and neoplastic cells? So one method was by releasing the cytotoxic granules full of perforin and granzyme, and the other method was that the Fas ligand on the T cell binds to the Fas receptor on the target cell, and that induces apoptosis. All right, that's it for the quiz. I'll see you next time.